Welcome to Stat's virtual event, the ASH Recap Live, sponsored by Janssen. Thanks for joining us. I'm Angus Chen, cancer reporter here at Stat. Before we get started, I have a few quick housekeeping items. At the bottom of your screen, you should see several icons. Use the chat feature to chat amongst yourselves or to send a message directly to the Stat team. Please don't put your questions for us in the chat. If you do have a question that you would like to ask us during the event, please use the Q&A feature to type your question, and we'll try to answer it live. In the Q&A window, you should also be able to see the questions that other attendees have asked and upload the questions you would most like to see answered. We'll be taking questions throughout this chat, so please don't wait until the end to submit. This is part of what makes these events fun. Thanks again to all of you for being here. And before we get started, I'd like to welcome Serge Masurlian, US President at Janssen, for a sponsored introduction of today's event. Thanks, Serge.
Thank you, Angus, and thank you to Stat News for the opportunity to kick off today's discussion. I have to tell you, what great energy to gather together at ASH. It was another meaningful ASH meeting. And, you know, we appreciate ASH for many, many reasons, but the most important of those is really that it gives us the chance to connect and to listen and to learn from each other, which I know all of us really, really value. So I think it's important also to frankly express our appreciation for what we all do in supporting patients and really working together collaboratively in partnership to fight this disease. You know, for me, ASH is a meeting that holds very special meaning. I started my career in industry about 22 years ago. And ASH was the first meeting that I had joined. It was actually 2 weeks on the job. I was, fresh out of grad school. And had taken a role in as a something like a sales representative, MSL type role. And frankly, I was hooked from the get-go. Really, the value of the scientific exchange and debate. And I distinctly remember, sort of walking down the promenade, having just met a KOL. It was the first time I was in, sort of exchanging ideas with a KOL. And I remember getting into a really interesting and frankly heated debate about gene therapy. And I know at the time there were a few gene therapy programs, and the KOL I remember was adamant, saying that within 3 years gene therapy was going to be the standard way to practice medicine. And it was kind of interesting because I had a little bit of experience with gene therapy at that point in time as part of my graduate program. And, I took the debate the other way. For me, I didn't think we had gene therapy licked at all. I told them, we'll be lucky that this thing works within 20 years. And so here we are 22 years later. I'm not sure quite yet that, gene therapy and or other novel therapies, cell therapies, et cetera, are the standard way to practice. I am very optimistic, though, in terms of what the future holds, and very excited to be part of this community, to be able to bring very novel and highly impactful therapies to the market. Now, what I remember also about that conversation with that KOL at the time was it really started to implant in me the value of scientific exchange and debate. And as we think about industry and industry partners as an example, the value that we bring to thought leaders, care providers, and the like, and the type of partnership that we build very much, I think, relies on being evidence-based, very much in terms of bringing value to each exchange and interaction, and scientific value at that. So I have to tell you, my purpose back then really hasn't changed. For me, what I've always been passionate about is, and still am, is really being passionate about advancing science, ultimately in the service of patients. And that's certainly a big part of my purpose. Collectively, I know that's a shared purpose that we all have. We're fighting to beat cancer together, really from bench to bedside, and everyone here has a critical role in that battle. At Janssen Oncology, our mission, as you can kind of see over me, is to reimagine care and redefine living. And that's really what inspires us every day. So, of course, our long-term vision for the organization is to cure cancer and work to achieve that. But that's the long road, I believe, still. We have a lot of work to do, a lot of partnership to do, but there is a lot that we can do today in terms of innovating and thinking creatively and how to be able to advance care and ultimately the experience for patients and the care delivery ultimately for those patients. So The patients fundamentally keep us grounded. It's partnerships like these, like with many of you, that we value working together ultimately in the service of the patient. It's really about continuing to look forward, right? And working with this cancer community to frankly allow us to help us achieve for patients, really the best possible outcomes by way of certainly survival, but also quality outcomes, the quality of life that they experience. And that's a very, very important part of our mission. We're really reimagining as part of our vision how to connect and collaborate differently with the healthcare professionals, the patients, caregivers, and other industry partners like yourselves. And we do this, across the care continuum, as I mentioned, care delivery, and really working to push the boundaries along the continuum. Our mission is really that, if we do this right, that patients can ultimately redefine living for themselves in their own ways that are meaningful. And that's what, sort of gets me out of bed every day. And I'm sure that's what gets many of you out of bed every day. And, during ASH, like I have to tell you, we were very excited to share some of our progress with you at Janssen, how we've come together in partnership with the scientific community, frankly, to answer some very important scientific questions. I couldn't be more proud of the organization, having had over 45 company-sponsored abstracts. Along with that, in partnership with investigators, we had 35 IISs that we supported. So it's very exciting. It's an exciting time. We're humbled, to be able to work in partnership with, so many of you and so many thought leaders, scientists, clinician scientists, and the like, frankly, to advance the paradigms of treatment in multiple myeloma, as well as in the CLL space, which are, two important areas that we focus on. Really, along these lines, we recognize that we're just one part of the broad scientific ecosystem. We cannot solve cancer on our own, right? That doesn't make sense. Our approach to R&D is pretty unique. And frankly, we're pretty agnostic as what the source of innovation is. And really, our job is to continue to identify and create opportunity, really to pave the way towards our innovation strategy. And frankly, we're agnostic as to where the great ideas come from. We're in search of the best ideas, ultimately. Whether they come from a university or a research organization or a biotech or another pharmaceutical company in partnership with academics, it really doesn't matter for us. What's most important is that we identify, qualify the best thinking and bring the best thinking and translate the best thinking ultimately for patients. So that's an important part of who we are at Janssen. Another important aspect of our organization is that it's, turning the best science into medicines. That's kind of one part of it. Our focus is also on obtaining the best outcomes, healthy outcomes, by means that go beyond the medicine itself. As you well know, and many of you are part of this, partnering with the oncology healthcare ecosystem, policymakers, professionals, advocacy groups, patient organizations. Our job is really to be able to create a very seamless experience ultimately for patients and for many who are challenged, by access, to be able to support access and to be able to close health equity gaps for people. We seek to learn and work alongside physicians and patient organizations to be able to develop both programs and tools to allow for ultimately better care, better communication, again, as I say, throughout the care journey, right? So the bottom line is that we're really about developing and delivering solutions to improve not just the quantity, right, of time on Earth, but the quality of that time as well, which is very, very important. So with that, I wanted to thank you all for your contributions. You've made such an incredible impact on the patient community, and we've done that together. Our work together helps us all do more for patients, and at the end of the day, that's really what matters most. So I hope you enjoyed today's event. I wanted to wish you and your families a very happy holidays, and I'm passing it back now to Stat. Over to you.
Thank you, Serge. Here to join me for the rest of today's discussion, please welcome Adam Feuerstein, Senior Writer for Biotech at Stat, and Dr. Jane Winter, Professor of Medicine, Division of Hematology Oncology at Northwestern University's Weinberg School of Medicine, and 2022 President of the American Society of Hematology. And last but not least, Michael Sekaris, Chief Division of Hematology, Sylvester Comprehensive Cancer Center, University of Miami Health System, and Communications Committee Chair of the American Society of Hematology.
Hey everyone,
welcome
how's it going?
everybody.
Hi there.
Hey everyone.
I appreciate both of you joining us today. Thank you.
Thank you for having us.
Yeah. All right. So I guess I want to start off by asking the two of you, and maybe Dr. Winter, you can answer first. But what was it like to attend ASH in person? You know, I mean, traveling to medical meetings used to be the norm, but we haven't gotten to do that for a while.
It was terrific. It was energizing. It was better than expectations. I think everyone was very excited. There was a hunger to be together. And I think for many of us, it really fulfilled a lot of that need.
What about for you, Dr. Sekris?
I like the use of that word, Dr. Winter, hunger. It had been 2 years since we had seen each other. And you realize that human beings are social animals. We really enjoy the interaction, and a two-dimensional Zoom interaction isn't the same as actually meeting people in person, attending a session, being able to lean in to somebody next to you and ask for their opinion on what's going on also, and try to figure out where you're going to settle out in interpreting the science. And you know, something that happened that was pretty special about this meeting. You would go up and see somebody you hadn't seen in a while, and just innocently say to them, "How are you doing?" And there were a lot of times someone would start to tear up. And these are hardened hematologists, right? These are folks who are used to emotional settings where we're talking to patients about really important parts of their lives. And you realize just how much we missed it, and how much probably a lot of us are all a little bit traumatized about what's happened in the past couple of years, and just how special an in-person meeting at ASH was, so that we could reconnect and also appreciate such in-depth science.
Yeah, I feel like I had a little bit of FOMO because I was home covering this, and Angus was in Atlanta and I was at home. But you could totally see that, Dr. Sekris, what you were saying through. I mean, I saw it through the Twitter feeds. I was following the hashtag, and I saw some of your tweets. I know you were reconnecting with people that you hadn't. Clearly, a lot of people were doing a lot of there was a lot of reunions going on. It seemed like in the halls of the meeting, and it functioned afterwards. And I mean, you could really tell sort of like the sort of the relief and joy that people were having, kind of getting back together.
The intellectual interchange, though, is also different. It's different when you're in the room. You've got to be in the room when it happens. And I think that there is a difference. And just having a discussion face to face in the room, have individuals getting up to the microphone, the level of intellectual exchange is really quite high and different from what you get on a routine Zoom call.
Yeah.
And I think part of that, it's another good point, Dr. Winter, is that one of the purposes of a meeting is that someone will present science, and then it's the job of the rest of us to actually be critical of that science and to be the gatekeepers for whether that science is something that's believable and should be advanced and should be incorporated into clinical practice. And it's so much harder to do that when you're interjecting on Zoom, as opposed to a meeting where you see people walk up to the microphone and see the line and see how much interest there is.
Yeah, I mean, it's also, I mean, I saw this a lot, which was, I don't know. I mean, it's something that can only happen in person, where like after a session, people would go up to the speakers or talk to each other, and as they're moving around, they'll have these kind of spontaneous interactions. And like I overheard one person just was talking about an experiment, and someone who he hadn't really collaborated with before was like, "Well, why don't you send me your reagent? I'll try it." Which is like that just kind of thing wouldn't happen on Zoom, but it is sort of what happens when you get to be in person with each other.
It's part of what makes science special.
Yeah.
Well, especially these days when I think many of us feel that science is not held with the same reverence that it once was. And so to see different presentations that have different results, the same kind of question being asked, and everyone trying to sort of understand differences between studies and hold them to high level of review, I think was very reassuring. This is the scientific process. This is how we go forward in making new advances, and particularly with the background of what is happening around us. I think it was very important to see that at play.
Yeah. Well, let's talk about some of the data that was presented at ASH this year. From our perspective, I guess, some of the really interesting abstracts were new data from the ZUMA-7 and TRANSFORM studies of axicell and lysocell, respectively. These seemed like they garnered a lot of attention and a lot of debate over whether they could change the way physicians treat patients in second-line B-cell lymphoma. So I'd love to kind of hear what your thoughts of this are. Are there any strengths or weaknesses, any kind of unanswered questions that you have, about these data?
I think the big issue is the Belinda trial. So that was a late-breaking abstract. And with Tisacell, the same kind of design, but there were clearly some major differences in terms of bridging or not bridging. And those results were not did not support the benefit of CAR T as second-line therapy. So I think that's where everyone is struggling to understand and look at the details and understand these differences. I think there are clearly some very methodologic differences that can explain the difference between the Belinda trial in contrast to ZUMA-7 and Platform, which were both supportive of CAR T for second line.
And what are those differences, Dr. Winter? What do you think? Do you sort of chalk it up to the trial design? Some people talked about manufacturing. It took longer to make
exactly
Kymriah.
I think that the problem, to my mind, we have to look very carefully at the details, but the time to infusion for me as a clinician who deals with this day in and day out, I need a product. These are the patients who were enrolled on these studies were all very high risk patients. So these were individuals who, and a third of them were double or triple hit patients. So these are patients who don't have time. There is no time to spare. And so that's key. And in the Belinda trial, the median time to infusion was almost twice. It was 50 some days, which is a very long time. And those patients went through a number of different regimens or cycles of treatment prior to. So I think that there were differences. ZUMA-7, steroids only for bridging, and the interval was very short, I think it was 29 days to infusion, so very different. And so I think the study design in permitting all that bridging and the time to, it's not clear to me if the problem was time to manufacturing or I specifically asked that, and I'm told that part of the delay was that investigators kept wanting to try additional cycles of treatment prior to as bridging. So I think there are lots of details that we need to study and
do you think that you need those details before you change the way you treat patients? I mean, that's always the practice changing question always comes up, when these kind of studies are presented.
Well, I think it's very clear that so you have to be very careful that these studies looked at patients who either progressed through therapy, they're primary refractory, or they relapsed within a year. So this is not for all patients. The data doesn't support those patients who relapse later, okay, who more likely can get through second-line treatment and potentially benefit from an auto stem cell transplant. So these are patients with very proliferative, high-risk disease, of whom, the very small fraction of patients who actually got to transplant, about a third of patients. And then the plateaus on those curves haven't really changed. They're not good. And so I think those of us who take care of these patients know that an auto transplant doesn't provide any to my mind, for patients who are double or triple hit. Pursuing that kind of strategy, that standard of care, is not likely to be beneficial. So I think we're going to be moving in that direction. We need the approval. This is an extremely expensive kind of strategy. It's labor intensive. There are going to be alternatives, I think. We're going to have off-the-shelf products. We're going to have more bispecifics. So there will be alternatives, but this is a big step forward, I think.
Now, while we're on the topic of CAR T, I'd love to get your perspective on sort of the use of CAR T therapy in the clinic right now and how it might be evolving. I spoke with one clinician at the conference who just very bluntly said that pretty much all the patients he's seen go on CAR T therapy relapse, and that it might be time to start thinking, about what's after CAR T, what kind of new therapies or new technologies might be used to augment the current CARs that we have.
Yeah, absolutely. I mean, the curve show only about 40% in the long term, but that's a big improvement over where we were previously. But clearly, we need to use this as a stepping stone. And I think we're seeing there's so much going on in terms of off-the-shelf products. If we didn't have to wait, and the expense is huge. And so we need something that is not a designer product, perhaps a off-the-shelf allo type CAR T. Or bispecifics. The problem with the bispecifics is they are administered on an ongoing basis, at least right now. So there's a lot to come.
Yeah. So there was I'm really curious to hear what you both think of this. But there was a plenary session on the association between CHIP, or clonal hematopoiesis of indeterminate potential, and Alzheimer's disease, a rather surprising association. I'd love to get your thoughts on this. If you yourself were surprised by the data that Dr. Jaiswal's lab at Stanford found.
I can jump in if that's okay, Dr. Winter. I think we were all a bit surprised by the finding, and I think that's why it's a plenary. CHIP, so clonal hematopoiesis of indeterminate potential, of course, is something that occurs across people's lifetimes and increases with age. So probably over the age of 70, 10 to 20% of the population has CHIP abnormalities. And these mutations, the most common of which are TET2, ASXL1, DNMT3A, or the DTA abnormalities, are associated with both evolution to myeloid malignancies, which is my specialty, and also associated with heart disease, which was a really fascinating finding, and then recapitulated in mouse models. So we believe kind of the common pathway linking heart disease to myeloid malignancies is inflammation. And the purpose of then exploring the association between CHIP and Alzheimer's disease is, well, maybe some of this inflammation is also linked to acquisition of Alzheimer's disease. So the investigators looked at some data, and specifically in different subtypes of Alzheimer's disease, some of which are less likely to predispose a person to developing Alzheimer's, and some of which are some of the results that can come back from the genetic testing people sometimes get for each other for holiday presents, with Christmas coming up, are more associated with developing Alzheimer's disease. And what they found in microglial cells, which are thought to be kind of almost this immune scavenger system in the brain for plucking out those protein deposits associated with Alzheimer's, is that there was less of a likelihood of acquiring Alzheimer's disease with these CHIP abnormalities. Now, certainly I would look at this as preliminary data. I mean, it's almost this, good news, bad news scenario, and probably the only good news we've had emerge from having chip abnormalities. If you live long enough to get Alzheimer's disease, you may be more likely to have Alzheimer's disease. So we'll see what the next steps are with it. But yeah, wow.
Yeah, what are the implications? I mean, what would you like to see done next with a study like this?
I just, Adam, there's just this part of me that wonders, okay, what do you do with this next? Like, what's the clinical implication of this? What happens with preventive strategies? So there are a bunch of us who have chip clinics that have opened up within our hematology divisions. The purpose being that there are a lot of people out in the community who are being diagnosed with chip and then don't know what to do. And this was particularly true as mutational testing was really taking off. I would get a few referrals from folks with, okay, this doctor sent this mutation panel, they told me I have TET2, what do I do next? And there actually weren't prescribed guidelines set up. So we've created these chip clinics both to be able to advise patients and also to start to engage in preventive strategies. For example, linking a hematology chip clinic with a high-risk cardiology clinic. We're now starting to see the emergence of clinical trials of interventions. For example, if you give high-dose vitamin C, can that reverse some of the untoward effects of having a TET2 abnormalities? Well, if we're doing that, are we now then potentially putting people at risk of developing Alzheimer's disease? And I don't think there's enough of a clinical implications of this or something that's out of the lab yet to make that connection. But I'd love to be able to see if there is really that connection so we can inform patients as they're in these chip clinics.
So there's two questions that came in, and one was sort of a jokey question, which I'll start with, and then the serious question. So the jokey question was, should we all be microdosing doxorubicin on a regular basis to avoid Alzheimer's? And then the more serious question is this just survivorship bias? Are you just looking at people who live longer, don't have chip, so they're more likely to age into getting Alzheimer's versus people, with chip more likely to have shorter lifespan?
Yeah. So with the first question, the low dose doxorubicin, I might worry a little bit about the cardiac effects of doing that. So maybe not. But with the second question, which is exactly the same thing, I wondered as well. This is it's a matched cohort, so presumably for some of these things, like age itself, they've been able to control for. But are you really controlling for almost a volunteer bias? So the people who contributed samples to these databases, were they more likely to contribute because their Alzheimer's wasn't as advanced, or they were younger, or what that was? So it's a very valid question.
I think the whole thing shows how important blood cells are, okay? And hematology has this far-reaching impact. And sort of we talk about off-target effects of drugs. I think these are the off-target impact of a lot of the work we do in hematology that we don't quite recognize yet how far-reaching some of our findings are. And that's why it's important to come together and have different points of view, different kinds of individuals, at the same meeting who have different thoughts. I mean, this is ripe for lots of discussion, I think. So I look forward to seeing where it goes.
Yeah, I mean, Dr. Sekaris, you mentioned, the emergence of chip clinics, and this is something that I've wondered a lot about, in sort of the field of clonal hematopoiesis, is like, what is like, well, first, like, I guess, how do clinicians feel about these chip clinics? Do they find it useful for their practice? Do they feel like it is benefiting their patients? And for the patients who are diagnosed with chip, I mean, is it frustrating for them to sort of feel like, well, I have this condition, I don't know what to do? Or is it the opposite? They actually feel more empowered with this knowledge?
I think the answer to all of that, Angus, is yes. So the number one question that I've seen when we've created, I've been involved in the creation of a couple of chip clinics, is, do you really want to do this? Do you want to focus this much attention on something that somebody has that may ultimately result in nothing untoward. But I really am a firm believer in knowledge is power, and I think placing those findings in context is really important for folks, at least at their initial diagnosis. So I've certainly seen patients in my practice where they were told, you have a TET2 abnormality or you have a DNMT3A abnormality, you're now at a much higher risk of getting leukemia, so make sure you follow with a hematologist, right? Terrible introduction to that, but something that really does happen. So then having a CHIP clinic where I can meet with somebody or one of my colleagues can meet with somebody and put that in context and say, you know what, the risk is higher, yes, but it's an absolute risk, and let's talk about, you know, what the real risk is to you over a lifetime and how we're going to manage this and what strategies can we put in place to maybe minimize that risk. For example, if somebody smokes, you can really now have an engaged conversation about stopping smoking. I think that's ultimately helpful to somebody. So I think it's, I apologize, Dr. Winter. I think as an established clinic, I think if you're not trying to drum up business by doing a lot of testing and diagnosing CHIP and then bringing them into a clinic, I think if you're managing diagnoses that have already been made, it can be very helpful, particularly if you tie it to some mutations that are detected that aren't somatic, they're not acquired, they're actually potentially germline. And then you've folded into a kind of a familial mutation clinic as well.
But part of all of this, we had a number of sessions at ASH that spoke to predisposition genes. So there was a workshop on Friday and additional sessions later in the meeting. So understanding genetic predisposition may be very important in terms of ultimately selecting therapies for patients when they develop alternative other diseases and solid tumors, for example. Do you want to take someone who has certain predisposition genes and give them this therapy versus perhaps another therapy, and the long-term consequences of that in the development of second malignancies? So it's all very exciting, I think, in terms of being able to look forward and perhaps design our treatments based on understanding the genetics of the individual that you are planning to treat, and when that will, become a means of, Mikhail, will you think that we're going to be doing that soon, looking at every, when a new patient comes down the road with a lymphoma, I need to know what their genetic predisposition is?
Interesting question. I mean, certainly there have been studies that have looked at patients who have breast cancer and those who had CHIP abnormalities at diagnosis, unbeknownst to them or their treating physicians, and then years later develop myelodysplastic syndrome that is believed to be therapy-related. It may be that those patients are at higher risks of therapy-related myeloid malignancies, but the treatment in and of itself isn't the sole offender that leads to those myeloid malignancies. It's probably they had a CHIP abnormality, and then this was a second or third hit that marched them on that road.
Right. Yeah.
I guess I'm really curious, since both of you mentioned sort of a bunch of potential sort of avenues that under sort of research on CHIP could go towards. Is there something that you'd really want to see this field lead to in terms of actions that could happen in clinic, and maybe more targeted screening, or maybe better precision medicine, like you were talking about, Dr. Winter?
Yeah, I think that's a great question, Angus. There was another abstract that was presented at the ASH annual meeting, a late-breaking abstract that looked at kind of the clonal dynamics. So how quickly does that clone grow over a period of time? Does that put you at increased risk for myeloid malignancies? That's another thing that can be done. I think it's a reasonable question in certain patients who are being treated for other malignancies, whether we should enhance our screening for CHIP. I also think it's a very valid question in those high-risk cardiology clinics to have the testing done from the opposite direction. Is it possible that one of those patients may be more at risk of a myeloid malignancy? And then we can start on the road of actually intervening to try to stop that from happening. There are clinical trials that have opened looking at interventions for patients with CHIP. Angus, I just don't think we're quite there yet, but we're almost there.
Okay. All right. Well, I want to, I guess, move on to another topic, the Agile and Panther trials. Dr. Seckers, I know you presented on the latter, and as a corollary, I'd love to get your perspective on presenting negative trial results. It's sort of the importance of learning from studies where the outcome, didn't really go exactly as hoped.
Yeah, it's an important topic, particularly in what I specialize in, which is myelodysplastic syndromes. I once heard somebody say that a negative study in MDS is the only instance at ASH where you can get both an oral presentation and a late-breaking abstract, because unfortunately it's been so common. So just to orient everyone, the Panther Trial was a randomized study in which we enrolled 454 patients with higher risk myelodysplastic syndromes, chronic myelomonocytic leukemia, or oligoblastic acute myeloid leukemia to receive either azacitidine monotherapy, which is the standard in these sorts of higher risk myelodysplastic syndromes in CMML, or azacitidine plus pevonedistat, which is a NED8 activating enzyme inhibitor. In these trials, most of the patients had higher risk MDS. That was really our target population. About 100 patients had oligoblastic AML, and then about 27 had chronic myelomonocytic leukemia. We had a couple of interesting findings from this. The first is that when we looked at CR rate and compared it in the higher risk MDS cohort between those who got azacitidine and pevonedistat and those who got azacitidine alone, the CR rate for those who got azacitidine alone was 32%. Classically, that should fall around 18%. So great news for the patients who were enrolled on our trial, bad news for the trial design, that they had such high remission rates with azacitidine monotherapy. When we looked at the trial was primary endpoint was to look at a difference in event-free survival. No difference in the intention to treat population at 2 months, but not statistically significant. In the higher risk MDS cohort, and again, that was sort of our target population, the EFS for the combination was over 19 months versus 15.5 months for azacitidine. For overall survival in the higher risk MDS cohort, the median overall survival was 21.6 months for those who got the combination versus 17.5 months for those who got azacitidine monotherapy. There was a trend towards significance, and that was an absolute difference of 4 months. And that 4 month, I want you to keep in mind for a second, because I think it gets to what we can take out of this. So primary endpoint of the trial was not met. This was a negative study, but what can we learn? And I think there are a couple of things. And if you have a negative study, Angus, what I try to really focus on is, okay, negative study, these are important because they help us build the next trial. And I want to take you back a couple of trials that led up to this that helped build this trial. The first was the North American Intergroup trial we conducted, in which patients were randomized to receive azacitidine monotherapy versus aza plus lenalidomide or aza plus the histone deacetylase inhibitor vorinostat. In that study, once again, no differences in the outcome. It was a negative study. That one was powered on overall response rates. But what we learned in that study was that patients discontinued the combination therapies prematurely or dose reduced significantly more commonly than in the azacitidine monotherapy. So then we used that information to build the pevonedistat trials, where we had very strict language about not dose reducing and not dose delaying for the therapies. And lo and behold, patients remained on these therapies for a median of 8 or 9 months, and you had a much higher CR rate in the azacitidine monotherapy arm, probably because they stayed on the drug long enough to appreciate a response. So we learned from that previous trial. This trial demonstrates that yep, keep patients on a long time, they're going to do better with aza monotherapy. And in that initial intergroup trial, the median difference in overall survival, not significant, was 4 months. The median difference in overall survival in this trial was 4 months. So one thing I tried to talk about when I gave this presentation is that I think we have to be bolder in myelodysplastic syndromes. Instead of powering studies to show a median overall survival difference in MDS of 6, 8, or 10 months, that's unrealistic for non-targeted therapies in a heterogeneous MDS population. I think we've got to power studies to show a median overall survival difference of 4 months in MDS, make incremental benefits just as folks have done in multiple myeloma, so that eventually we're getting to that 8 or 10 months.
I wanted to follow up because I think Angus mentioned the Agile Trial, which is well, I know it as Tipsovo, the brand name, and I think what was interesting there was it was a positive study, but also I know there was questions raised about, the changing standard of care in that setting and sort of what that meant. Can you maybe go through that a little bit for us?
Sure, I'd be happy to. So, in the Agile study, which was conducted, I believe, largely in Europe, patients with acute myeloid leukemia who were older, so unlikely to tolerate induction chemotherapy, were randomized to receive azacitidine monotherapy or azacitidine plus ivosidenib. All these patients had IDH1 abnormalities. So this is really a remarkable study given a very rare clonal abnormality. They enrolled 146 patients to this study, randomized them to receive the combination of ivosidenib and azacitidine versus azacitidine alone, and were able to demonstrate 3 times the CR rate for those who got aza and ivosidenib, and 3 times the median overall survival for those who got the combination versus azacitidine monotherapy. And these are really, really impressive differences, and ones that we don't see very often in older adults with AML. Interestingly, the CR rate for azacitidine monotherapy in these older adults with AML was 15%, very similar to what I told you before has classically been the CR rate for azacitidine in higher risk MDS. These are biologically very similar types of cancers. So there was some differentiation syndrome in those who received the IDH1 inhibitor, as there has kind of been described in the past. But this was, in my mind, for a limited patient population with a rare somatic mutation with AML, a wildly positive study. The question, Adam, as you mentioned, is, okay, that's great, but the standard of care is to give azacitidine and venetoclax in older adults with AML, which has shown a survival advantage versus azacitidine monotherapy. So what do you do in these populations? There were some studies that are starting to add targeted therapies to the azacitidine plus venetoclax backbone. Preliminary stuff, kind of phase 1. There was a whole session at ASH dedicated to it. Showed very high overall response rates in just about every study that was presented in the session, as is kind of typical of these earlier phase combination trials. We're waiting for now randomized versions that's now going to compare those triplet combinations to azacitidine and venetoclax. But you may ask, where would I see a role for azacitidine and ivosidenib given these data? I will tell you, the combination of azacitidine and venetoclax is not easy to give in older adults. And there are a bunch of us who would argue that azacitidine and venetoclax is closer on a continuum to standard 7+3 chemotherapy than it is to azacitidine monotherapy. And the way the label is written by FDA doesn't quite align with how we actually have to give this in practice. We have to dose reduce the venetoclax because we also have folks on antifungals that interact with the venetoclax, and then we have to continue to dose reduce it or truncate the duration of the venetoclax so patients can tolerate it. So I would look at the azacitidine and ivosidenib combination as something I would give to older adults with an IDH1 mutation who probably are frailer and maybe much older, so not just over 60, but over 75, over 80, or have comorbidities, who I think probably would have a rough go of it with azacitidine and venetoclax.
Mm. And then sticking with AML, but another targeted therapy, I know there's been some interest in the Menin inhibitors. I think there was an update on SNDX, I think 5613, this meeting. What's your perspective on those drugs?
Yeah, they're intriguing. It's for a pretty select patient population. So those who have either MLL abnormalities or NPM1. MLL is relatively rare in AML. NPM1 is not. The update in the study enrolled 54 patients, showed a very complicated list of CR rates. The absolute CR and CRH was 22% of 45 patients who were evaluable. Then you start to add in kind of softer CRs, and those start to take on a little less and less clinically meaningful relevance. But a single agent in patients who received a median of 3 previous treatments for their AML That's pretty strong data for a single agent so far.
And there's another compound that I know was put placed on clinical hold because I think it's because due to differentiation syndrome, and you mentioned that. And I know this is something that you see in these patients. I mean, what are your? I don't know if you have any thoughts about that, or you're aware of what's going on, and whether that's kind of an issue to look for moving forward.
Yeah, I think if you have a patient who has been exposed to, 2, 3, 4, 5 previous regimens, and you're now considering a drug like this, in all honesty, I think that person should be seeing a leukemia expert. And folks who focus on this very rare cancer subtype like I do are pretty used to dealing with differentiation syndrome or tumor lysis syndrome or cytokine release syndrome or any other badness that drugs can cause. So it doesn't worry me as much in the right hands. Is that a fair response to that?
All right, so I want to move on to another topic, which specifically is bispecific antibodies and their role in treatment, especially compared to CAR T cell therapy. These seem to be the two sort of lines that people have been really excited about for a few years now. And specifically, there were some new results on mosunetuzumab in follicular lymphoma.
You did that well, I guess. That's a hard one.
It is. These drug names are always a mouthful. But I would love to hear your thoughts on that study and just generally the development of bispecifics in combination therapy or as single agents.
I think it goes along with the whole theme of immunotherapy and seeing how T cells can be very effective in and of themselves in treating disease. The mosunetuzumab study was, in follicular lymphoma. This is a disease where, the vast majority of our patients do really well with a median survival of, as I always tell patients, in decades. So the newly diagnosed patient is going to live 20 years and probably, if we update the database, probably far longer. But there's a small group of patients who do poorly, who relapse early after treatment. And those are the patients I think that were being addressed there. It's in contrast, we now also have good outcomes in follicular lymphoma with CAR T, which is a single effort, a single treatment, and hopefully I'm keeping my fingers crossed of my patients who we treated on study who remain in remission. But the bispecifics are highly effective in terms of responses, but they need to be readministered. It's not a one-time strategy. The other aspect about them is that they are, as Mikhail says, some of us feel very comfortable. There is CRS. There are same kinds of toxicities, albeit they are low grade. They're grade 1 or 2. But in the mosunetuzumab study, they about 8 or 9% required tocilizumab. So, we're giving toci to patients. This is not someone who's just going to go home and, lives 2 hours away from me and so on. So I think that there's a lot of work to do in figuring out how these are going to fit in. Are they a bridge to something else? Because our patients with follicular lymphoma, they want something durable. And, in general, this is a disease that keeps coming back. What's durable? An allo transplant, not something very applicable to, the vast majority of patients are not willing to go that way. Once upon a time in my career, I did that. I would take young patients with follicular lymphoma to allografts. But now we have so many different alternatives. We seem to recycle. We go from one category to the next, and lots of combo clinical trials. So I'm not yet sure how this is going to all shake out, but I think it's proof of concept that that T cells, and whether it's a BiTE or a bispecific or CAR T, are very durable. The one thing we haven't talked about is we had the Beutler presentation from Margaret Shipp and Steve Ansel on checkpoint inhibition in Hodgkin lymphoma. I know Hodgkin lymphoma is not as common, but the outcomes there with checkpoint inhibition are pretty dramatic in Hodgkin lymphoma. And my group, we had 2 of the presentations in the Hodgkin lymphoma session, updating our data with upfront pembrolizumab in previously untreated patients with Hodgkin lymphoma, showing we continue at 100% progression-free survival now with nearly 3 years of follow-up. And also, the checkpoint inhibitors are showing incredible activity in combination with chemotherapy in our relapsed patients. So it's another area in which we're using immunotherapy and those T cells to dramatically improve outcomes for our patients.
Dr. Winter, I want to actually just follow up on the durability question, particularly as it relates to allogeneic CAR T approaches, because the durability issue has kind of been the thing. It's a question that a lot of people have, whether these are going to be durable enough, and then there's some thoughts about redosing. Taking an off the shelf CAR T and then being able to redose it. What are your thoughts about that?
I think there's a lot of, we are fine. There's a lot of fine tuning, and even more than fine tuning that remains to be done. But I think we have concepts and recognition that this category of therapy is effective. I'm sort of thinking that our CAR T, we're sort of hitting a there's much to be done. We're not really, if we have 40% plateau there in our CAR T for large cell lymphoma, for follicular lymphoma, we're doing better. Mantle cell lymphoma, we have incredible outcomes, but how durable are they going to be? But surely we're going to be able to figure out hopefully an off-the-shelf kind of strategy, so that we're not, we don't have to be manufacturing these personalized products for patients that are so expensive, that the applicability becomes limited. I mean, we're going to be hitting a wall in terms of the cost of our therapies, which I think is a very important issue to be addressed with some of our news. So we heard about the POLARx study now with the antibody drug conjugate as part of upfront therapy in diffuse large B-cell lymphoma, polatuzumab. And so we've got a lot of new frankly, if we can cure people up front, we're not going to have to be so dependent on all these relapse strategies. And I really think that's where we should put all our, let's cure them all. For every group of patients, we don't have to come back and make a CAR T cell product, or maybe addressing immunotherapy as part of upfront treatment, which is what I've done in Hodgkin lymphoma. And if we can cure people up front, then we're much better off.
On that topic, there were a lot of presentations this year on sort of screening for myeloma, which I know is not something that's done in typical sort of standard clinical practice. But one sort of really huge study, multiple myeloma from University of Iceland, presented the first data from a 75,000 person study, looking at ways to screen potentially for multiple myeloma. I'd love to hear your thoughts on this, Dr. Sekaras. I know you work on myeloma, so what do you think?
So it's a high compliment to say I work on myeloma, but I actually don't. I work on myelodysplastic syndromes. I leave myeloma to the smart hematologists. But I am aware of this study, which is really remarkable work of population-wide study just with remarkable size, right, and implications. Not so generalizable to the United States, right? We're talking about the population of Iceland as opposed to our population in the U.S. But I think it has implications for other cancers, and that is, what are you going to do about it? So, if you look at kind of epidemiology textbooks that talk about screening, there are kind of certain rules for why you should screen for cancers, right? One is that you detect a cancer at an early stage. Another is that you have a test that doesn't introduce a lot of side effects to a person, so it's low risk. The third is that you identify a cancer at an early stage that you can do something about so that it doesn't become a later stage. So if we're screening for multiple myeloma at a very early stage, are we going to intervene at that point? It's almost like the chip argument, right? Or MGUS, or are we not going to do anything about it? And therefore, is it worth the screening? And again, I'd have to defer to my myeloma colleagues about that, whether they would actually intervene at an early stage. And that takes a little bit of equipoise, because certainly there's industry support for intervening at an early stage and getting folks onto a drug very early on. And this is some of the critiques I've heard about smoldering myeloma trials, right? Why start people so early? It's certainly something that's been refuted in chronic lymphocytic leukemia, with a study it's decades old, and then a reboot of a similar study more recently that shows that intervening in CLL before you absolutely have to probably isn't benefiting patients in the long run. So
with follicular lymphoma, there have been generations, every decade, another study to show that there's no benefit to jumping in early, and particularly in a disease that has such a long-term, it's going to be around forever. So patients live with it. It's an issue of getting to learn to live with it psychologically for many of these patients.
We are unfortunately just out of time. So I think with that last thought, we'll wrap up here. Thanks again to Adam, Dr. Sekaris, and Dr. Winter for joining me today and for answering these great questions from our audience. And thanks again to our event sponsor, Janssen, and to all of you out there for taking the time to join us. I hope you guys all have a wonderful holidays ahead, and stay safe and be well.
And ASH next year is in New Orleans, I think. So
yes,
we'll see you guys there.
Looking forward to it. Please plan on being there in person.
I'm there next year, definitely. Thanks all.